200,000 tons. 200,000 tons of food are thrown out every day just in the United States alone.
Organic waste is also a major challenge for long-term space travel. Not only does it take up valuable storage space that could be used for other materials, but can also cause odor and air quality problems. In addition, organic waste can provide a breeding ground for bacteria and other microorganisms that could have a detrimental effect on the health of astronauts. According to NASA, the average human produces approximately 1.5 kilograms of waste each day. This is a significant amount of waste that needs to be managed and stored properly in order to maintain a safe and healthy environment for astronauts on long-term missions.
Hi, my name is Praneeth,
And my name is Roshan.
and we're seniors at Valley Christian High School. And this problem served as our inspiration to create our microgravity microbial fuel cell and test it aboard the ISS.
But first of all, what is a microbial fuel cell?
Well, a microbial fuel cell is an energy conversion device that uses microorganisms to convert organic matter into electrical energy. Microbial fuel cells work by using the metabolic activity of microorganisms to break down organic matter, such as plant waste, into electrons. These electrons can then be used to generate electricity. But you might be asking, how did we send this thing to the ISS? It's not like anyone can just call NASA and send their project to space, right? That's where the Quest Institute ISS Research Lab program came into play. In the ISS Research Lab, teams of students design and build microgravity experiments under the tutelage of experienced mentors. Through our partnership with Space Tango and NASA, we were given the unique opportunity to send our experiments to space. Together, we led a team of 8 students on our microbial fuel cell experiment. Due to the size and varied talents of our team members, we decided to split up responsibilities into biological, mechanical, electrical, and software. During the initial research phases of our experiment, all members were involved. However, once that stage was completed, each member took on their specific responsibilities and eventually combined their results to produce the final outcome. However, our mentors also played an important role in the success of our experiment. Mr. Amika Okikiocha, Mr. Howell Ivy, and Mr. Sandeep Shah served as our head mentors. That meant that literally everything we bought or did went through them first. They prevented us from making safety mishaps and, more importantly, unnecessary purchases. Initially, we were at a loss when it came to the research phase of building a microbial fuel cell. None of us had any prior experience with this type of project, so we had to start from the basics. We diligently read numerous research papers to gain a deeper understanding of the subject matter. Our mentors also suggested that we reach out to experts in the field for guidance, so we sent emails to over 100 microbiology professors. Eventually, we were fortunate enough to receive help from Professor John D. Coates from the John D. Coates Lab at UC Berkeley. He not only pointed us towards 2 valuable research papers on microbial fuel cells, but also offered his expertise by advising us on our initial design, and he even provided us with colonies of S. onidensis MR1, the bacteria used in our experiment.
But before we could actually start on constructing the cell, we had to do test after test.
In fact, we had 2 sub-experiments running in unison.
The first challenge was to decide the design and materials for a microbial fuel cell, considering the multiple ways to build one. We eventually landed on a solid design. We planned to have 2 chambers, an anodic and cathodic chamber. The bacteria would be housed in the anodic chamber, and the cathodic chamber would house the ferrocyanide. The 2 chambers would be separated by a semi-permeable proton membrane. Throughout the process of the experiment, sodium lactate and tryptic soy bath would be pumped into the anodic chamber to feed the bacteria. Finally, both chambers would be externally connected using a copper wire. The second challenge was figuring out how to lyophilize or hibernate our S. onidensis bacteria. Prior to sending our experiment to NASA, the bacteria needed to be in a hibernating state in order to survive the launch of the ISS. Lyophilization is an expensive and complicated process, requiring the bacteria to be tested with different materials, called excipients, to see if they can survive the freeze-drying process of lyophilization. This is why we contacted over 5 labs and companies, asking if they can lyophilize our bacteria pro bono. We received rejection after rejection, until finally, LyoHub, a company based in Purdue University, graciously agreed to lyophilize our bacteria for free. With a month left on the clock to submit our experiment, we sent 2 racks of test tubes to LyoHub for lyophilization. Luckily, it turned out that our bacteria love being lyophilized, along with whey powder, allowing us to use an S. onidensis whey powder lyophilized biofilm for our experiment.
So with our testing complete and our materials at hand, we got to work constructing our cell.
Despite our difficulties with finding professors and lyophilization services, our crowning disaster was when we dropped our completed experiment as we were turning it in. 3 days before the actual deadline. To our chagrin, both of our water bags filled with tryptic soy broth and sodium lactate burst open upon impact with the ground. However, we were determined to see our experiment through. We repurposed our MFC chamber and electrical circuitry and spent 10 hours a day over the weekend working on replacing the water bags and pump tubing. Through both stress and struggle, we managed to rebuild our experiment in just those 2 days. And as we were submitting our experiment right on the NASA deadline, we were extra careful in handling the chamber, making sure that we wouldn't suffer another devastating pitfall.
Over the course of a month, our automated experiment ran on the ISS, collecting data on the voltage production of the fuel cell over time.
It took another 2 months for us to receive back our experiment along with its data. The results were, quite literally, electrifying. The results supported our hypothesis as our fuel cell's voltage production increased over time whenever sodium lactate was pumped in, as compared to previous voltage peaks, indicating that as the S. honeidensis biofilm grew, it underwent anaerobic respiration more efficiently, thus producing a greater voltage across the cell. This production would not stand to last, however, as there was an insufficient amount of growth medium to sustain its growth past 6 days. With these results in hand, our team created a research poster and traveled to the American Society for Gravitational and Space Research Conference to present our experiment. To our happy surprise, we were awarded first place in the high school research project category, crystallizing for us the amount of work that we had placed into our beloved microbial fuel cell, and rewarding us for the struggles we had to go through to build our experiment to fruition. In conclusion, our team's research shows that microbial fuel cells are an exciting prospect in the realm of space travel. With their ability to generate energy from organic matter, they can help reduce the amount of space taken up by waste in outer space, while providing an efficient long-term energy source. By investing in microbial fuel cell technology, space explorers can ensure a cleaner, more sustainable future for space travel.